All right, so our presentation is about differential gene expression. It's an analysis in maize, tomato, and field mustard under drought conditions. This is our intro. So first of all, what is a drought? A drought is a deficiency in precipitation over an extended period leading to a water shortage. Drought is a slow-moving natural disaster that has moderately affected about 20 to 30% of the world's land mass at any time since 1950.
Why is drought an important area of study? The initial effects of a drought may be difficult to identify right away, so it may take weeks or months to determine a drought has started. The end of a drought is hard to identify for the same reason. Models predict that drought will become more frequent and severe as temperature rises.
So what does drought do to plants? In general, drought as a stressor decreases the photosynthesis and respiration rate of plants. It results in mineral and nutrient deficiencies, and it is also the most widespread factor affecting agricultural production.
All right, so looking at our specific crops, first we have the maize crop, which is corn. The drought effects on maize was negative, resulting in smaller plants and premature flowering, as well as possible death. And prolonged period of drought can genetically affect the seedlings and cause the next generation to also be affected.
When looking at drought effect on tomatoes, drought stress due to sufficient water supply was considered as the most destructive abiotic stress, which could happen at any crop growth stage and ultimately cause growth yield loss. In this study, significantly increased melatonin concentration was regarded as a response signal in tomato to apply stress.
Drought effect on Brassica Rapa. The second most important oilseed crop is a recent addition to the Brassica family. Very susceptible to drought conditions due to the low genetic diversity. Our research, we studied the effect of drought on a few important crops by examining how it affects the plant's genome and what genes are expressed. We also analyzed the genomes for upregulated or downregulated. Methods, we use cybers to run the RMTA, and we use the NCBI databases to find genomes, and we ran a differential sequence analysis in RStudio on cybers.
This is our TSEC analysis.
So when looking at this downregulated gene in tomato, the volcano plot shows the x-axis, shows the full change in gene expressions, and the y-axis is the negative log p-value. The blue on the volcano plot represents the upregulated gene, and the red also represents the downregulated gene, and the green is not significant. Also, when looking at this downregulated gene in tomato, under the drought condition, is this gene significantly down regulated and codes for protein involved in defense response and protection against the mildew infection. When looking at the upregulated gene, this plays a critical role in the plant defense against microbial pathogens as well as in reproduction and tissue development.
So the downregulated gene in field mustard, it is significantly downregulated and is responsible for sulfur uptake and acts as a sulfur deficiency recognition gene, meaning that under drought conditions, the plant won't be taking as much sulfur that's available in the soil due to a lack of water. The upregulated gene in field mustard is related to a disease resistance protein. It is responsible for cascading an immune response to pathogens and wounds, as well as signaling cell death to occur in a compromised cell or when natural cell senescence occurs. So under drought conditions, its immune system is compromised and it signals for more compromised cells to die when they need to.
All right, so the downregulated gene for the maize crop is a dehydrogen protein. This gene is specifically related to drought actually because it reacts to water stress and general stress, and also regulates leaf expansion and formation stages. So it directly relates to drought because it's directly relating to water stress. And then the upregulated maize crop gene is an uncharacterized protein, but relates to drought because it works with the breakdown of proteins and peptides through enzymes, and also works with the 2 and 3 leaf expansion stages, which are related to growth. All right, for the conclusion, we got this table that shows the amount of significant up and down regulated genes that change due to the droughts. These are compared to the total number of up and down regulated genes, and are also compared to the total number of genes overall. You can see that the maize crop had the greatest number of significantly upregulated and downregulated genes, which suggests it was the best to adapted to handle drought conditions compared to the other crops.
All right, and that's our presentation.